2 more minutes.
2 more minutes, oh yeah, now 12. But your clock and my clock are synced.
No, I just looked at my watch.
Oh, well, you have a watch. Mine's computed by. Oh, you have analog.
No,
oh,
I have
okay.
digital
well,
someplace too. Where did I put that? I do have this too. 12, 13.
Yes. Okay, so and now I'm finally getting the knack of doing this. After my 3rd year, I'm just beginning to feel I'm kind of getting it together. Well, how will I be 30 years from now? I think I'll have it down by then. Okay, so I'd like to introduce Garri Davydyan, and he's going to be talking. He's from Ottawa, came all the way here from Ottawa, Canada, and he's going to speak on split-quaternion representation of a functional hierarchy of biological systems. So let's give him a hand.
Biological systems, complex objects, and network traditionally used to describe system functional mechanisms. We can measure systems outcomes, which are physiologic parameters, and can find some input-output relationships between different systems. We may collect some information about internal components of systems and subsystems. For example, function of cells depends on inclusions, organelles. Functions of organs are determined by specific cells and links to other organs. The system, for example, cardiovascular system may include not only heart and vessels, but also nervous and endocrine systems as functional components. Shown links just indicate specific contribution made by other systems and subsystems. Another approach to the complex system is based on finding universal functional patterns or invariants regulating functions of biologic objects. Biologic objects, for example, a human body, are organized in a way that each morphological level consists of integrated elements of the previous levels. Separable and reproducible elements such as biological cells, biological tissues, organs, etc. are considered as functional systems. Biological systems not static, but developing objects. Development of species and individuals occur through the splitting of some characteristic features. Splitting increases the number of independent, autonomous parameters and the spectrum of systems' responses on environmental stimuli. Formally aligned elements of a homogeneous one-dimensional space becomes represented by points of the plane. For example, 2 branches come out from one stem on phylogenetical evolutionary tree. Functional elements and related characters which undergo splitting should be autonomous. To obtain new characters, also autonomous, some functional features of the previous level should be transformed by splitting mechanism to the next functional level. Formally, the splitting mechanism should be provided by an operator which is a direct sum of two operators with opposite eigenvalues to provide dynamical stability of the system's outcome. The operators genetically included in a regulatory system should be traceless. Here's an example. Evolutionary, the system controlling viscosity of the blood has undergone splitting into clot formation and clot degradation subsystems. Each of these subsystems also has some distinguishable components, which are known elements of biochemical cascades forming and destroying thrombi. By finding relationship between different biochemical components within each subsystem, we can build a network, most probably between some components. Functional relationships also exist. What kinds of functional links between systems besides network webs are known? There is only three types: negative feedback, positive feedback, and reciprocal links. These patterns provide stable functional relationship between systems. Slide shows negative feedback loops between pituitary gland and thyroid gland. Each system has its own output: pituitary, well-known TSH, thyroid, thyroxine. Reciprocal links between clot formation and clot degradation biochemical cascades. This slide was shown before. Clot formation system produce clotting factors to increase the viscosity of the blood and stop bleeding. Clot degradation biochemical pathways prevents clot formation and dissolve thrombi. And the last one, positive feedback, is found between uterus contraction and amount of released calcium by posterior pituitary gland. It also exists as a pattern regulating rectal emptying. Outputs for the first system are amount of calcium and contractile forces. For the second system, they are the strength of contraction of smooth muscles of rectal wall and degree of relaxation of anal sphincters. All three are reproducible and because of that should be genetically encoded in chromosomes. NPR functional elements have been established not only between organs but also inside biological cells. So based on biological and clinical observation, it was proposed that negative feedback, positive feedback, and reciprocal links, NPR abbreviation, form a universal functional basis for the internal structure of separable biological objects, namely biological systems. On the slide, there is a matrix representation of PNR patterns. Below are the images of PNR as operators of corresponding dynamical systems used in ordinary differential equations. Each trajectory describes functional regulations between two subsystems. Because the trajectory lies on the same energy level, each two-element system is autonomous. Matrices of this functional invariance are the same as basis element of Lie algebra special linear group over real numbers. Because of the Lie algebra representation, NPR patterns form three-dimensional linear space of regulatory elements of autonomous biological systems. This means that acceptable functional patterns can be obtained as linear combinations of the basis element of this space, and they will remain within the closed additive group structure of the Lie algebra as L2R. Multiplication of algebra element is not closed operation on the algebra, and it leads to quaternion structure with a non-commutative ring formed from four basis elements, vector part of which is represented by NPR patterns. The scalar part presented by unit matrix determines divergent process and can be considered as a component of destructive environment affecting autonomous functions of the system. From now, a functional structure of a system will be presented by operators or matrices of special quaternions or their vector parts. With a given norm of an operator as a square root of that absolute determinant value and a scalar product, which is a trace of a product of elements, split-quaternion will represent a semi-Euclid four-dimensional space having indefinite matrix signature. Splitting of species, characters, or local parameters during evolution or individual development is crucial in understanding what functional patterns determine each functional level and how a hierarchy is formed. The NPR space of regulatory elements contain one-dimensional isotropic subspace, which in case of split-quaternion basis has two pairs of eigenvectors over R and Ri with opposite eigenvalues. Splitting occurs in three directions to form I, J, and K basis components for the next functional level. Each basis element either a circle or hyperbola. Resultant surfaces are pseudospheres with real top and imaginary radii. During structural evolution, initially homogeneous space having chaotic interactions of containing the space element undergoes splitting on four 1D orthogonal elements or two orthogonal pairs of reciprocal because of opposite signs of eigenvalues components. Each blue dot is a one-dimensional measurable character representing systems output. During evolution or individual development, a system or character splits into two reciprocal subsystems, characters. Each reciprocal pair is covered by 3D space of functional elements spanned by NPR bases. The graph represents the final genetically encoded result. The splitting is reciprocal because initiating the splitting operator is a direct sum of two operators with opposite eigenvalues. Next slide gives more functional details. Each dot is one D character over R, which splits into three different pairs of orthogonal characters corresponding to 1D or 2D invariant subspaces over R. Obtained six-dimensional functional structure is a vector part of split-quaternion. Each component Si has two opposite eigenvalues either from R or Ri, giving rise for three-dimensional regulatory space of the next hierarchical level. Dynamical model of the obtained functional structure is a direct sum of three second-order ordinary differential equations. They give three-phase trajectories localized and orthogonal to each other planes. Obtained surfaces are direct product of circles and hyperbolas. Phase trajectory for the normal system will lie on these surfaces. The next level of more differentiated elements organized in a whole functional structure will be a split-octonion. Its dynamical model is a direct sum of two split-quaternions, or two pseudospheres. As a whole system as a regulatory structure, split-octonion is fairly hypothetical in modeling biological or medical objects now. The eigenvalues of a diagonal 2 by 2 matrix are factors of determinant function. It corresponds to a value of quadratic form obtained from the split-quaternion matrix representation of 2 by 2 matrices over real numbers. Because there is no one-to-one correspondence between one-dimensional operators of the previous and four-dimensional ones of split-level spaces, the eigenvalue as a single factor of determinant of 1 by 1 matrix will cover the whole surface of pseudosphere. As a regulatory structure, split-quaternion gives certain flexibility or certain range of normal acceptable conditions of the integrated upper level, integrated level. There are three graphs representing a functional structure of the two-element system depending on considered level. Below the graphs of two-element systems, there are supporting them matrices having the same block matrix view as the basis, positive, negative feedback, and reciprocal links patterns. There are no medical or biological examples for split-quaternions. Formally, if this pattern as a whole functional unit exists, it should be genetically encoded, and metabolic transformation will lose associativity as elements of split-quaternion ring structure, as follows from just mathematical structure of split-quaternion. And the last slide, functional regulation of gastrointestinal tract elements. So there is three different colors: stomach, large and small intestines. On macro level, these organs have distinguishable morphological features. They even grow differently. Each intestine can be presented as a sequence of functionally separable portions or tube segments having at the ends not anatomical, not always anatomical, but functional figures. The relationship of neighbor portions are regulated by reciprocal links. Contraction of proximal applicated portions occurs together with the relaxation of the following portion. The same mechanisms coordinates functions of different organs, for example, stomach and intestine. This type of regulation is known as gastrocolic reflex. It may have a functional structure of split-quaternion function, inside of which intestinal segments, all three types of R and P regulatory elements are supposed to exist. There is no direct information. The functional element you obtain from ordinary differential equations. The same regulatory structure has cardiovascular system. Another example is clot formation and clot degradation cascades, which are regulated by reciprocal interactions. Each cascade is a sequence of biochemical transformations. Both are relatively autonomous parts regulated by P and R patterns. If so, the rheology or viscosity of the blood on molecular level is regulated by split-quaternion structure. If it's genetically fixed in chromosomes, it should be shown. So it's just theoretical. But strictly following from existing data and just mathematical logic, there's nothing else. Not all facts just prove, but many of these facts really take place.
Okay, we still have 4 minutes, but are you
Thank
finished?
you.
Yes, I think.
Yes.
Thank you. Because everybody is tired.
Okay, that was really interesting. And does anybody have any questions here for Garri? Yes.
So I wasn't sure why you excluded the positives on the diagrams from being biological.
Actually, it's a good question.
It
Yes,
seems like
I
they're
excluded
just
and
unrelated to
I'll
each other.
explain why. Environment is something coming outside. It always exists together. And this is the reason why split-quaternions as a structure is used in these explanations. But we can just take it out And all what's happening inside the system depends on only 3 components. Other components makes just physical impact, not structural. It's like microbes. They can affect your body or other body, and we can eliminate microbes. Other work our body can do by itself. But on the other hand, cancer is different. It's not always possible that our body can eliminate this because it goes inside the structure, inside these 3 elements as a basis components. It's not only 3, it's actually the whole other type of groups, but genetically fixed 3, in my opinion.
Yeah, it's a, you know, I listened to your work, and it's the second time I've heard it, and so it's just a comment. I mean, it's bold to sort of say, you know, in some deep way that there are only these 3 things. There's some positive feedback, some negative feedback, and these reciprocal links, and somehow these things provide some deep biological structure that is sort of impermeable to whatever else happens.
On each level, this structural hierarchy actually is one of the types of hierarchical structure.
Yeah. Well, so a quick question, you know, in terms of the literature, what are there any other people who are working in this area who have a similar sort of theory about the way in which? I mean, it's extremely interesting. I mean,
to convince people that it can work because it's actually together mathematics and medicine and biology.
I mean, have you published any of these results anywhere?
I'm trying.
Yeah, okay.
But it's hard.
To publish this.
Because it's, you know, people like lab work and statistical results of lab work. But theoretical papers, people very skeptical and acceptable. Yeah, it should go from, you know, what scientists, whatever.
Okay.
Everybody should know.
Any other questions?
Yeah, could you explain the last slide, if you can put it back up?
He's already unplugged.
Yeah, the computer's off. That's your computer.
Yeah, could you explain the slide again, especially like the reciprocal links part?
Reciprocal links?
Yeah.
They exist between, for example,
transitions
large
between the random
intestine and small intestine, the big parts of intestinal system. And they also exist within each part. What is interesting regarding large intestine, they have, how to say it, structure which is actually anatomically also fixed. In small intestine, we don't have any sphincters inside. We have functional sphincters. They're working, they're squeezing 2 parts of intestine when it's needed in order to separate the segment. But in large intestine, we have actually separated its gausters. In Latin, it's gausters, and they have actually this separable structure, but it's practically not well known. It's long time ago, 60, 70 years ago, in good anatomical atlas, it was explained that they have some things like that. So, and inside, it's also working as a reciprocal, because 2 segments, 2 neighbor segments, when one is collected,
It's contracting.
hand,
Expand, contract.
other squeezes in order not to let it go down. But when first segment started to contract,
expand.
others, so it's reciprocal.
Okay.
But between stomach, for example, and large intestine also exists reciprocal, but it's not exactly reciprocal actually. It's reflexes, so it's probably collection of combination of other things. And actually rectal emptying also very interesting actually, and very not simple structure from physiological, surgical, and clinical point of view. One of the hardest part of our body, not heart. Heart is simple. This is hard. because it's working in different direction. 3D, it's not about it. It's different horticulture.
Thank you.
Okay, thank you very much. Any,
Thank you for this opportunity to tell about this, to talk about it.
well, I think that actually Garri has been with us now for, I think, all 3 quaternion sessions, special sessions that we've had. We started the special sessions actually 3 years ago, and this is our 3rd one, and we hope to continue it on for another 30 years. Okay, so it's wonderful to see everyone here, and now we have a couple of more minutes before our next speaker. And did your students, are your students
Yeah,
here?
yeah.
Okay,
Speakers.
great. And do you need a computer? Do you want
I've
to?
got my computer.
Okay, so we can set up.
Do you have a remote we could use?
Oh, yes.
Wait a minute.